Seeing new products on the market can be exciting. New foods may be marketed with various benefits and claims. However, eating something new, something you've never tried before, can be scary. We are comfortable with what we know when it comes to food. Either way, the food needs to be safe. Cell-based food, also known as cell-cultured or cultivated food products, are an example of new food products. We visited a production facility for cell-based food and saw how cells from a cow can become a steak. Several scientists walked us through the different steps used in producing cell-based steak.
This building in Rehovot, Israel is home to a food production facility. Up a few floors from the ground level is where workers gather to produce cell-based steaks. Producing food from plant or animal cells is a new form of agriculture. The first phase of cell-based food production starts with a living animal, a cow in this case. A veterinarian checks the cow for diseases to be sure it is in good health. The cells are extracted, then transferred to the production facility.
Hello, I'm Ronit Bakimer. I'm Aleph Farms VP of Quality and Product Safety. We are now in Aleph's analytical lab. Cellular agriculture in general and culture meat specifically are made of cells. These machines allow us to look at the cells and to make sure that they comprise the characteristic that we're interested in along the process of production.
One of the tools we use for the analysis is a flow cytometer. So the flow cytometry is based on technology which detects specific markers for the cells, and the detection is done using a laser beam. And here we can see that the majority of the cells, or majority of those dots, are positive for a specific marker.
A polymerase chain reaction, or PCR, is run to check gene expression level. This is one of the steps in the cell line development to make sure the cells are healthy and will perform in a consistent manner in production.
Additional tool we use for analysis of our samples and the final product is the PCR. So the PCR is intended for analysis of RNA levels in our samples, and RNA represents gene expression in each sample that is tested.
We start from deriving the cell line from the cow in a way that was used for many years by cattle breeders. We characterize these cells, we see how they grow, if they are appropriate for our production process. We can see here how the cell look like when we grow them.
What you can see here on the monitor is an immunofluorescence assay which we use to mark our skeletal muscle cells, which express specific skeletal muscles protein. You can see the skeletal muscles in red.
We can look at the microscope here and see that the cells look healthy. They grow very rapidly, they divide, and they are appropriate to serve as our source for the cultured meat product. And when we characterize these cells and determine that they are appropriate for establishing cell bank, we freeze many of these cells in these kind of vials, many of these vials, and we call this a cell bank. Here is where we keep our cells frozen in liquid nitrogen in a very low temperature of -195 Celsius. They are all frozen in this kind of vials, and now I'm going to open it, so you'll see, and I have this for protection. We have all these boxes inside. In each of them, There are many of these vials. We can keep here many vials like this, and it can be kept for many years. Each vial from the working cell bank that is thawed will be a source for hundreds of kilograms of meat. The advantages of a cell bank is that it is well characterized. We test the cells and we see how they grow and how they behave, and we assure that our process is reproducible. So every time that we thaw these vials, the cells are growing the same and produce the same product that we expect. And another advantage is that we can test these cells and make sure that they are free of any contaminants like bacteria or viruses.
As we continue to scale up, we introduce methods and equipment for high-throughput analysis. Here you can see an example, a robot. This robotic system enables us fast and accurate analysis of our bovine cells and cultivated meat products.
As in any other food, food needs to be appealing, food needs to be nutritious and tasty, but the first and utmost important thing is the food safety. And the same for cultivated meat. We use the approach of using only safe ingredients and following the safety of the production process for early R&D stages all the way to pilot and scale up. In this vessel, we prepare the cell's growth media. This is a 1,000 liter container. After it's been ready, it's been mixed and ready, we take the media and filter it through this filter to make sure it is sterile and safe for the cells. Then we hook it here and it goes to the production room. When we're done filtering, we take the filter and make sure it's intact. So it's complete and filtration was efficient. Our safety approach is, I would say, I can divide them to two. One of them is looking at the intrinsic safety of the product, of the process, and the other one is the operational. In the intrinsic, I would count the inputs, that is the cells and the growth media. In the operational side, we work according to good manufacturing practices. We work according to risk-based considerations, where we go along the whole process and we define what are the potential hazards and we mitigate them before they will be introduced into the process. This way we make sure that when we get to the final product, it's clean and safe.
So right now I'm setting the parameters for my tissue bioreactor, TBR for short. The bioreactor mimics a living body of the cow itself. So right now I have my suspended cells on the scaffold, which are nourished by the medium, which you can see right here in our big vessel. The vessel provides all the building blocks that are needed for the cells to grow and to nourish and become a steak afterwards.
In this part of the process, the air cleanliness is very important. The air is filtered through filters in the ceiling and enter into the room. We have two tools to measure the cleanliness of the air. One of them measures the presence of bacteria in the air. We set a plate, an agar plate, in the machine, and air is sucked inside, and if there are any bacteria in the air, they will grow in the plate. The second one is the other equipment. This sucks air into the machine and it measures the number of particles in the air. We should be at a certain level according to the cleanliness we define for this room.
In addition to air, the water needs to be clean.
Purified water are used throughout the production process. It gives us the ability to control the inputs into the process, while the purified water contain no minerals, for example. We start by taking city water, which are good for drink. But we continue purifying them, removing elements from them. At the end of the process, the purified water are stored and circulated to all the users in the facility that they use them for their production. The water goes through pipes, which once a week go through a sanitation process using high temperature. The water are constantly circulated in the loop, so they're not settled, which prevents bacterial growth. In addition, the cleaning procedures of the water also make sure that there are no bacteria or any other elements that would reside in the water before we use them.
We are sampling samples from our water system and we analyze them for total organic carbon, which is a parameter for quality of the water. We put it in this instrument, which is about 15 minutes. We're getting the results, and we're monitoring several sampling points during every week or every period of time. This is very important for our production.
So the challenges of moving from small-scale to large-scale production relates to the sterility of the production process. We have to make sure that the same sterility that we have in small scale in the R&D laboratory, where we work in biological hood, can be transferred to the larger scales in bioreactors, in the plants themselves, and while transferring cells from one vessel to another vessel.
After the cells have adhered and grown on the scaffold, the product is harvested from the bioreactor.
So once we finish with the process itself after a couple of days, then we'll start harvesting our steaks. Once we harvest the steaks, then we'll have to wash our tissue bioreactor. And we'll wash it in 2 consecutive cycles. The first one is washing with water and base, which we clean the entire vessel and the pipes surrounding it. And the second cycle is the sanitation one, which we use steam in 120 to 125 degrees Celsius in order to sanitize it from any remaining bacteria or any other bioburden left inside the vessel.
Inherently, the production process for cultivated meat cannot incorporate any pathogens. If some pathogens are incorporate the culture of our mammalian cells, they would take control over the culture and will not allow our meat cells or animal cells to develop. So our meat is inherently pathogen-free. At scale, we plan not to use any antibiotics, which is also another aspect related to antimicrobial resistance. And last but not least, because our process occurs in one single facility from A to Z and is fully monitored and checked at every step, we can ensure a full traceability and transparency of the process, with the documentation of any test performed and the ability to provide the full set of data on the meat we produce and deliver to the consumers.
In addition to making sure the product itself was produced safely, we also need to make sure that the environment in which it's being produced is clean. And what we will see here is we take a swab of the machine used to process the food, and we make sure there's no residues from previous production or any bacteria or anything else. Now we got a low level of contamination, which means it's clean, as expected. Every morning before start of the shift, we clean the surfaces in the working areas to make sure it's ready for a new working day. And the last part of the production, we take the final product in its wrapping, in its packaging, in the primary packaging, and put it in a vacuum machine, which will make sure the product stays safe. Now a vacuum is being created inside. All the air is being sucked out of the bag to make sure no environmental bacteria are left inside and no oxygen is inside the product to keep the shelf life. Now there's no air inside the bag, and the product can be stored for a long period of time. Temperature control is another important factor to keep the product safe. The room we're now at has low temperature, has only 15 degrees Celsius, which we keep throughout the processing of the final steps of the processing of the product. In addition to that, when we finish processing the product, we insert it into a shock freezer, which immediately drops the temperature down to minus 15 degrees for long storage. To make sure the product is safe, we do hazard analysis and identify the critical control points that may put hazard for the consumer. One of them is potential presence of small items that may cause physical damage to the consumer, like small metals from the equipment. The metal detector is our way to detect that there are no metals in the product. No metal was detected in the product.
The product is ready to be cooked and eaten.
Have it, a sneak preview of a new product. While the process for producing cell-based food is new, the 4 phases of the production may already be familiar to you. Like other forms of agriculture, cell-based food first needs to be sourced, like a seed or an egg. Then they can be grown until they are ready for harvest and processing. Safety needs to be first and foremost in every step.